Alright, so now we're going to start talking about antigen processing and antigen presentation. And then we'll transition into talking about T cells, natural killer cells, and B cells in more detail. Alright, so here we have our antigen presenting cells. Alright, so our macrophages. Our B cells and our dendritic cells. Alright, so our macrophages are going to be phagocytic and also present antigens. Your B cells also will process foreign antigens and display those antigens on their cellular surface as well as produce antibodies. Your dendritic cells are basically just antigen presenting cells. That's it. That's their main role. Alright, so looking at antigen presentation. Alright, so we have MHC class 1 and 2. Alright, well, MHC class 1. Alright, this is going to stimulate your cytotoxic T cells. Alright, so your cytotoxic T cells are your CD8 T cells. Alright. And they interact with cells that are displaying MHC class 1 surface markers with foreign antigens. Okay. So this is going to trigger a cytotoxic cell-dependent response. Your MHC class 2 molecules, right, are going to be perceived by interact with helper T cells, right, also referred to as CD4 T cells. So keep in mind that your antigen presenting cells, right, your macrophages, your B cells, your dendritic cells. Alright, these guys have both MHC1 and MHC2 that they can present on their surface. Right. All other cells of the body that have a nucleus only have this MHC class 1 surface marker.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class 2 MHCs, and the two structures combine. The MHC class 2 antigen complex is transported to the plasma membrane. The displayed MHC class 2 antigen complex can stimulate other immune system cells to respond to the antigen.
Alright, so looking at MHC class 1 and 2 antigen presentation. Alright, so your MHC class 1 antigen presentation are going to be antigens, in this case, intracellular antigens. Alright, so you're talking about a virally infected cell, or you're talking about one that contains a intracellular bacterium. Alright, so in this case you're talking about endogenous antigens inside these cells. Alright, and these antigens are processed and they're presented on MHC class 1 molecules. Now, keep in mind that endogenous antigens can also be things like components of worn out organelles, cellular structures, internal cellular structures. Alright, so those would make up your self-antigens. Alright, so keep in mind that these MHC molecules, alright, they're used by the cell to display normal self-antigens as well as foreign antigens. Alright, so this is how they communicate with your T cells. Alright. Now, your MHC class 2 surface molecules. Alright, these antigens are going to be exogenous. Alright, so this involves your phagocytic cells. Alright. And these phagocytic cells engulf foreign antigens, foreign cells, right, like bacteria, for instance, and they'll process the bacteria by, the normal phagocytic route of taking that vacuole and fusing it with a lysosome and breaking down the cellular components, and then those components get displayed on MHC class 2 surface markers. Right, so those processed antigens are displayed on MHC class 2. Alright, so this figure is giving you a little bit more detailed description of what's going on when these antigens are processed. Alright, so here, your MHC class 1, you're dealing with endogenous antigens. Okay, so you have your endogenous antigen that's degraded. Alright, so let's say it's a protein. Alright, so you have a protease that cleaves that protein into individual peptides. Alright, so short sequences of amino acids. Alright, those are going to enter the endoplasmic reticulum, right, where they're going to undergo processing and be attached to MHC class 1 surface receptors, and then those surface receptors, those MHC class 1 receptors, are going to be transported to the plasma membrane and displayed on the surface, along with your antigen. Now, your MHC class 2, you have your phagocytic cell. It phagocytizes your little bacterium from the extracellular environment. You have the fusion of that phagosome with your lysosome. The internal components get destroyed. Right. You break down that bacteria into its individual peptides and carbohydrates and lipids and nucleic acids. And then you have your little MHC class 2 surface marker. And you have your antigens that get attached to your MHC molecule, and then that vesicle is then transported to the membrane where your antigens are presented. Alright, so here you're being shown in a little bit more detail antigen presentation and the interaction between helper T cells and MHC class 2 surface receptors and MHC class 1 and cytotoxic T cells. Alright, so here you're looking at your little phagocytic cells engulfed a bacterium. It's basically degraded that bacterium internally, and it's displaying antigens of that bacterium on its surface through MHC class 2 molecules. And here your little helper T cell. Your helper T cell has a T cell receptor with a variable region, right, that's specific to this particular antigen. Alright, that's specific to that particular antigen and that specific epitope on that particular antigen. So this interaction will stimulate this T helper cell to then differentiate into, and we'll talk about this in a little bit, Th1 or Th2 helper cells. And we'll talk about what responses you get when you're talking about Th1 and Th2 helper cells. Okay. Now you're also going to have an interaction between your cytotoxic T cells. Alright, so your cytotoxic T cells also have a T cell receptor. Alright, so it has a constant region that's actually going to be inserted in the membrane of the T cell. And then you have this variable region which attaches to your antigen, to the epitope on your antigen. Alright, and then your MHC molecule, MHC1 protein, alright, you have your little constant region that attaches into the membrane of your antigen presenting cell, and it's displaying the antigen and the particular epitope that your T cell is interacting with. Alright? And its variable regions are specific to this particular epitope. Alright, so this is showing you, kind of taking a step back, the overall picture of this interaction between your antigen presenting cell and your T cell, cytotoxic T cell, interaction between class 1 MHC surface molecules and processed antigens with your T cell receptors. Alright, so basically this antigen presenting cell is telling this cytotoxic T cell, hey, here's an antigen, you need to be on the alert, you need to basically undergo clonal expansion and migrate to the site of infection to destroy any infected cells. Here we have our little helper T cell, right, that is being stimulated by a antigen presenting cell. So our class 2 MHC molecule is being displayed, and this surface marker, you have your antigen that's triggering your helper T cell to either differentiate into a Th1 or a Th2 T helper cell. Alright, so Th1 is pro-inflammatory. Alright, this will actually stimulate a cytotoxic response. Alright, Th2, the opposite. This is more of humoral immunity. Alright, so this will stimulate your B cells to, if they've come into contact with the same antigen, to undergo clonal expansion and differentiate the plasma cells and secrete antibodies. Alright, now we'll talk about what's going on in more detail between this interaction and the chemical messengers that are released and the big picture a little bit later on. Alright, so this kind of gives you a big picture overview of antigen processing and presentation by MHC class 1 and 2. Antigen presenting cells, or cells that present antigens using MHC class 1 surface receptors or MHC class 2 and 1 surface receptors. Alright, so you'll notice there's a similarity between your MHC class 1 protein molecules. Right? They have a portion that sticks in the membrane, and then they have a variable portion that actually is presenting the antigen to your T cell receptors. Now, you'll also notice that your T cells, so your cytotoxic T cells, alright, along with this T cell receptor, they also have this CD8 co-receptor. Alright? The CD8 co-receptor helps to, one, stabilize this interaction between your cytotoxic T cell and your MHC class 1 presenting cell. Alright? So, this also is going to serve as an extra trigger, if you will, because the CD8 receptor interacts with this MHC surface marker. And this helps to basically give the appropriate signals to the CD8 T cell that, hey, alright, this is an appropriate interaction, alright, this antigen is being presented, alright, you need to be on the alert, you need to undergo this clonal expansion and produce more T cells. Your MHC class 2 antigen presenting cells, right? The T cells, along with the T cell receptor, have a CD4 co-receptor. All right, it does the same thing as the CD8, except the CD4 is specific to your T helper cells, and it's a co-receptor that helps stabilize this interaction between your T cell and your antigen presenting cell, and also serves as co-stimulatory molecules to help trigger the response between this interaction between your antigen presenting cell and your T cell. Alright, so which of the following is not an antigen presenting cell? Alright, so we have macrophages, we have dendritic cells, we have T helper cells, we have B cells, or are all four of these antigen presenting cells? Well, in this case, your T helper cell is not an antigen presenting cell. Your T helper cell responds to antigens that are presented by the cells that are displaying antigens on their surface through MHC molecules. Alright, the T helper cells, your T cells, don't process antigens themselves. Alright, they respond to antigens. Alright, so we've talked about so far the two types of T cells. Alright, cytotoxic and helper T cells. Alright, so helper T cells are involved in stimulating humoral immunity. Helper T cells are also involved in some groups of helper T cells. are involved in a pro-inflammatory response and secreting chemical messengers, chemokines, that trigger other cells like macrophages or other T cells like CD8 T cells to become activated. Your cytotoxic T cells kill virally infected, bacterially infected host cells. Alright, so your Th1 T helper cells, alright, release these cytokines. These cytokines promote inflammation. Alright, these chemical messengers also trigger your CD8 T cells to become activated. They also trigger your antigen presenting cells to become activated. Alright, your cytotoxic T cells are responsible for destroying virally and bacterially infected cells. Alright, and they use enzymes called porphyrin and granzyme to do this.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class 1 MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class 1 MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
Alright, so here are cytotoxic T cells. The cytotoxic T cells are interacting with virally, bacterially infected host cells. And these host cells are displaying on MHC class 1 these particular antigens. Alright, so through MHC class 1 presentation, antigen presentation, your T cell receptor binds to that antigen being presented. Your CD8 co-receptor interacts with this MHC molecule, giving co-stimulatory responses to trigger this T cell to begin secreting two enzymes. One called porphyrin, the other called granzyme. Alright, so porphyrin creates these little holes in the membrane of the cell. And then granzyme enters the cell, basically degrading proteins, which ultimately result in the cell undergoing programmed cell death or apoptosis. Now, along with this, you can have other signaling molecules from your CD4 Th1 T helper cells. Alright, these chemical messengers will also activate your cytotoxic T cells to begin secreting these enzymes. So it's kind of a co-stimulatory reaction. Alright, so there are a lot of these control measures in place. There's a lot of these secondary reactions that are needed in order to trigger this cytotoxic T cell to do its job. Alright, because you don't want your cytotoxic T cell just destroying any host cell. Alright, you have to have the appropriate interaction and the appropriate antigens that are foreign that are being displayed, that are actually foreign and not self. Alright, so here this is showing you this interaction of the CD8 T cell destroying the bacterially infected cell. Alright, so you have your pores that are formed through your porphyrin proteins, enzymes. You have the influx of other molecules in, like granzymes, destroy proteins. These holes in the membrane also destabilize the cell's ability to regulate its osmotic potential. Alright, so ions and water entering and leaving the cell. So it's highly susceptible to lysis as well. Alright, so CD8 T cells are programmed to kill all the following except what? Cancer cells, virus-infected cells, bacteria, foreign cells grafted into the body. Alright, so in this case, your cytotoxic T cells are mainly going to target virally infected, bacterially infected, foreign cell grafts. Cancer cells are basically your natural killer cells, your NK cells. That's the job of those guys, mainly. Alright, so your Th1 helper cells, helper T cells. Alright, so these guys are pro-inflammatory. Okay. So you have your infected cell, where you have your phagocytic cell that's displaying MHC class 2 surface antigens, and these MH2 class surface molecules are displaying antigens to your T helper cells. And these T helper cells can differentiate into either Th1 T helper cells or Th2 T helper cells, as well as memory cells. Now, an activated T helper cell will actually secrete a chemical called interleukin 2, which will activate itself. It activates the T helper cell and also activates other T helper cells. Now, this T helper cell can differentiate into a Th1 cell, which is pro-inflammatory. It'll interact with a macrophage, your phagocytic cells, and stimulate this phagocytic cell through chemical messengers to become active. Or a Th2 cell, T helper cell, will interact with B cells to allow for antibody production. Alright, so this Th1 response, you have the release of pro-inflammatory cytokines that stimulate your antigen-presenting cells, your phagocytic cells, like your macrophages, to become increasingly phagocytic, basically increasing these cells to become competent.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T-cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T-cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells.
Alright, so your Th2 helper T cells stimulate humoral immunity. Alright, so here you have your B cell. Your B cell is also a antigen presenting cell. Alright, so you have your B cell that detects some surface antigen through this variable region of the antibody. That antibody is brought into the cell, the B cell, processed. And those antigens are displayed on MHC class II surface receptors. Now, for some types of antigens, like for instance, we'll talk about this in a little bit, certain repetitive antigens like carbohydrates, this will stimulate your B cell to produce plasma cells and memory B cells. But other antigens that are a little bit more complex, like proteins, require secondary interactions to help activate this B cell. Alright, so this is where Th2 helper T cell comes into play. Alright, so here we have an antigen presenting cell that has processed an antigen. And it's displayed on MHC class II surface receptors. And you have this T cell that interacts with this antigen that's being presented by class II MHC. It differentiates into a Th2 T helper cell. This Th2 T helper cell receives the appropriate signals to undergo clonal expansion. And one of these clones comes along and interacts with your B cell through its MHC class II surface receptor and the T cell receptor on the T helper cell. You also have your CD4 co-receptors helping to stabilize this interaction. And now, with the interaction of this T helper 2 cell, it will receive the appropriate chemical signals, these cytokines, that will trigger your B cell to become activated to differentiate into plasma cells and memory B cells. So that B cell will now undergo clonal expansion. It will differentiate into plasma cells and begin creating antibodies, differentiate into memory B cells, and be present in the blood for an extended period of time. Alright, so this kind of helps summarize your Th2 T cell response. Alright, so T cell dependent antigen stimulation of B cells. Alright, so you have your antigen presenting cell, your macrophage, displaying the antigen on its surface through MHC class 2. You have a T cell, a TH, you have a T helper cell, CD4 co-receptor, interacting with this antigen presenting cell. Alright, so you get the correct stimulatory molecules, alright, secreted by this macrophage, pro-inflammatory, pro-interaction, that's stimulating this T helper cell to secrete interleukin 2 and other chemical messengers that activate this T helper cell and also activate other T helper cells. Alright. To activate cytotoxic T cells, also activate B cells.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
Alright, so here are natural killer cells. They're targeting a cancer cell, secreting chemicals that ultimately cause that cell to rupture and lyse. The rupturing of the cell membrane and damage of DNA and undergo apoptosis. Alright, so here you have a normal host cell. Alright, natural killer cell interacts with that normal host cell. That normal host cell is displaying an MHC marker. and everything's normal. Cells that are virally infected or cancerous tend to not display MHC on their surface. So the lack of this MHC surface molecule on these diseased cells, the natural killer cell won't detect its presence. And these cells also will display stress proteins. And these natural killer cells have surface proteins that interact with stress proteins on host cells. So it'll receive the appropriate signals that is interacting with a host cell that's infected or cancerous and trigger that cell for destruction.
Antigens are normally processed by antigen-presenting cells and presented to T helper cells on class II MHCs. Only the T helper cells with T cell receptors capable of interacting with the antigen are stimulated to produce cytokines. The activated T helper cell stimulates only B cells that react with the same antigen, thereby limiting the response to the production of antibodies that were presented. Superantigens, such as the toxic shock syndrome toxin produced by Staphylococcus aureus, are not processed by antigen-presenting cells. Instead, they bind directly to the outer portion of an MHC class II antigen on antigen-presenting cells and to the outer portion of the T cell receptor of T helper cells. Rather than binding to only the cells specific for that antigen, which represent about 1 in 10,000 T cells, the superantigen binds without antigen specificity to as many as 2% to 20% of the T cells. This results in stimulation of many T helper cells. The result is the release and activity of excessive amounts of interleukin-2, IL-2, and other cytokines that enter the bloodstream instead of only acting locally as they normally do. This leads to symptoms of fever, nausea, vomiting, diarrhea, and sometimes shock, with failure of many organ systems, circulation failure, and often death.
Alright, so these superantigens, for example, toxins, some toxins, will bind to the T cell receptor and the MHC2 surface receptor, and cause this prolonged interaction between your T helper cells and your MHC class II cells. And so your T helper cells begin secreting these pro-inflammatory cytokines. And it's this over-amped up production. So you get what's referred to as a cytokine storm, all right, which can lead to toxic shock or shock. Alright, so you get an overactivation of T cells. So rather than this small localized response, you get a huge systemic response, which is something you don't want. Alright, so this is a picture of toxic shock syndrome. So this is very similar to what can be seen in patients that exhibit septic shock. All right, you see a lot of the same symptoms. All right, so septic shock is due to your bacteria in the bloodstream that begin multiplying to extremely high numbers, and you have this example, gram-negative bacteria have the endotoxin, the LPS, that triggers the septic shock. All right. Toxic shock syndrome is due to a superantigen. All right, something like toxic shock toxin 1 produced by like Staph aureus. All right, so this particular toxin was problematic for a while. All right, for example, for women, because of the type of tampons that are being produced. And based on the absorbency of these tampons, you had this microenvironment that was conducive to the overpopulation of these Staphylococcus aureus cells and the overproduction of these toxic shock proteins, toxins, that stimulated toxic shock syndrome. So you see the little petechial hemorrhages on the surface, which is showing you that there's leakage of blood fluid from the vasculature into the surrounding tissues. You see the systemic across the body, so purpura. You have the actual sloughing off of skin. Alright, this is another response to this particular toxin. You're seeing the skin actually sloughing off. Alright, so for each of the properties listed, determine if it describes a T or a B cell. Alright, so it matures in the bone marrow. Well, that's going to be a B cell. High numbers of cells circulate in the blood. Well, in this case, you can have B cells that are circulating in the blood usually. Your T cells, you're going to find them in your thymus, your spleen. It requires antigen to be presented with MHC molecule. Alright, so your T cells require antigen presentation to be stimulated. Produces antibodies. Alright, your B cells. And then cells activate to help other immune cells suppress and kill abnormal cells. Those are going to be your T cells. Alright, so these are your cytotoxic and T helper cells.